Today I'm joined by Laureate Professor John Aitken, who is a global andrology expert, co-developer with ASX listed Memphasys Limited of the Felix device and newly appointed Research Director with Memphasys. John, thanks for your time.
Good afternoon, David. Thank you for inviting me.
Well, you're no doubt excited by the commercialisation of the Felix device, which is, for those who don't know, a device that utilises a technology known as electrophoresis, combined with size exclusion membranes to select the best quality cells for improved IVF treatments. We're here today to talk about the other technologies and research you and your world-class team at the University of Newcastle in New South Wales, Australia, are working on for Memphasys. You're working on a portfolio of novel reproductive biotechnology products targeting high-value commercial applications. What is the general focus of this work?
Yeah, so we do run quite a large laboratory. It's one of the largest reproductive science laboratories in the country. And the focus of the work is not exclusively, but largely on male reproduction. And one of the reasons for that is that we know so little about male reproduction. Our knowledge of female reproduction is probably 20 years ahead of the male. And there are a number of problems in male infertility, and that applies to both clinical medicine and also to animal breeding. So we are focusing on trying to understand how spermatozoa are produced, how they function, and on the basis of that knowledge, devise new techniques for isolating these cells and for diagnosing infertility when it arises, and also preserving these cells. So these are major targets for us in our research group.
Memphasys's most advanced novel product is its Samson Stallion fertility diagnostic tool, which continues to progress through the prototype development phase and has been shown to perform effectively in lab trials. While its initial use is in natural breeding for thoroughbred horses, another application could be for quality control checks of sperm quality for AI in non-thoroughbreds, as AI is permitted for their breeding and for other species as well, such as cattle. Why is Samson important and what is it?
So in the thoroughbred breeding industry in particular, artificial reproduction is not allowed. So all conception is by natural cover. Within the business, it's very important to know whether a given cover is likely to result in a pregnancy. You'd like to know something about the quality of the semen that was inseminated at that moment. So what Samson is a simple device which can be used in the breeding sheds in order to assess the quality of the semen that's been inseminated. Essentially, we recover some residual semen after the stallion's dismounted. We put that residual semen into a device, literally press a button, and it provides a readout which tells you whether that was a good quality sample that was inseminated. So it's a simple diagnostic. And I guess where it's different from most other diagnostics is that it's been constructed so it can be used in the field. It can be used in the breeding sheds. It can be used in the doctor's surgery. It's not, as we normally have these kind of diagnostics, something that has to be sent off to a laboratory for a group of experts to do laboratory tests on. It's a simple one-stop test.
And it's important for those industries you mentioned because time is of the essence.
Indeed. So there are two things there. One is that you want to develop devices that people in the industry will actually use in the field. So you don't have to be an expert in reproduction to use these devices. All you need to do is collect the sample and put it into the device and it will give you a readout. And it is very quick. So instead of doing if you're normally, when you're assessing the quality of a male animal, you would do a microscopic assessment of the semen and you will count the number of cells that were there. You will classify the shape, the morphology of the cells and study their motility. Well, all of those measurements are wrapped up into one single reaction, which literally takes 10 minutes to resolve at the end of the day.
And you say it's a simple device, but a lot of technology and research has gone into the creation of it.
Yes, I mean, really, there's a lifetime of experience and knowledge about how sperm cells work that's gone into the development of the device, and in particular into the underlying chemistry. What is the chemical reaction that we use to understand the quality of semen? So yes, it's the product of a very long journey. But the fortunate thing is that you don't need to have gone through that journey to be able to use the device. It's a very simple device, but gives you a readout which I think will help people in the field, animal breeders, know whether a given insemination is likely to result in a pregnancy.
And animal breeding, as we all know, is a very lucrative business. How watched do you think this technology will be by the breeding industry?
Oh, I think if we can, so right now the Samsung device is in clinical trials. And if we can demonstrate that the readout of this device does give an accurate reflection of fertility and the likelihood of conception, then it would be something that the whole AI industry would be interested in, very much so. And that AI is used predominantly in cattle and horses, but also in other domestic animals like pigs as well. So yeah, I think it will be a game changer for the industry if we can make this work.
And how confident are you that you can make it work?
Well, so in the case of Samsung device, we have validated all of the underlying biochemistry and done laboratory tests to prove that this single reaction will predict both the number and the quality of cells in a suspension. So in a laboratory setting, I can be 100% confident that it works. The challenge will be, well, that's okay, but does it work in the field? When you're actually in the breeding sheds and there's people using the device that are not skilled in science, don't know much biochemistry, can you make it work under those circumstances? That's going to be the real test. But the underlying principles, 100% certain that this actually does work. It's a very powerful technology we've developed. And the trick will be translating that into a practical setting.
And in terms of practical setting, you are looking to test it in the current breeding season?
That's correct, yes. So we've been slightly delayed by COVID, but nevertheless, we're confident we will get into clinical trials in late October, early November. And that will be ample time to get enough information to determine whether the readout of the system is giving us a good correlation with pregnancy outcomes in the field.
Now, in conjunction with the University of Newcastle, Memphasys is also developing a technology to enable human semen to be held at ambient temperature and shipped from the donor directly to andrology and IVF laboratories for sperm quality assessments. Why is this important, and how advanced is this technology?
Yeah, so it's important because a knowledge of andrology, male reproduction, is not widespread. There are very few andrology experts in Australia, for example. And so the expertise to be able to really look at and study human semen and get a prediction of whether or not it's going to be fertile or not is in very limited supply. So a new business model is set up whereby people are establishing centralized diagnostic laboratories, which have all of the equipment and expertise that you need to diagnose male fertility. And to make that work, you have to have systems for shipping the sample from the site of production, from the doctor's surgery, or even from the patient's own home to the centralized laboratory for analysis. And this really came about because I was contacted by 3 independent companies, one in North America, one in Europe, and one in New Zealand, Australia, who are all developing this business model and wanted a method for transporting semen at ambient temperature. So no freezing or chilling or anything like that. You just put it into a transport device and send it to the diagnostic laboratory. And it will be unchanged for a period of about 7 or 8 days is our target. So that's in quite advanced stages of development now. Again, we've got really good proof of principle data. We are doing lots of laboratory work on that at the present time. And I'm very confident we will come up with a device and a medium that will enable us to preserve samples, semen samples, for long periods of time. And the beauty of that work is that the underlying principles will be translatable to other species. So imagine how it would revolutionize the AI industry in cattle if you didn't have to freeze samples, if you could just ship them to the point of insemination with a courier or just in the mail. This would, and the same for horse breeding, it would just revolutionize domestic animal practice. So the first cab off the rank is the human, because the market for such a device is immediately there. But once that technology is developed, I'm very confident it will find application in domestic animal markets as well.
Now, you're also working on new sperm separation devices, particularly for ICSI.
Yeah.
Why is that important?
Well, ICSI is, so first of all, what is ICSI? So ICSI is a technique which is used for, traditionally used for very poor quality semen samples, where the sperm cells, even if you put them next to an egg, couldn't achieve fertilization. And what you do to give them a helping hand is you physically inject the sperm cell into the egg. And it's a very effective way of getting conceptions in human, but other species as well. Several horses have now been generated using ICSI, and the same with cattle. It's a very good technique if you've got low numbers of poor quality spermatozoa. The problem with ICSI is that, unlike life, the gamete that's used for fertilization, the sperm cell that's used for fertilization, is not highly selected. So in life, in our species, for example, some 200 million sperm cells start a journey, and only about 50 cells will actually get to the egg, and only one of those cells will get into the egg and fertilize it. So each one of us is the product of a highly selective process. But when it comes to ICSI, you just pick a random cell and stick it into an egg and hope it's okay. So there is a demand then to make sure that when we're selecting cells for ICSI, they are the highest possible cells, quality cells we can get. And what we're doing is developing refinements to existing sperm isolation technologies that will enable us to, if you like, to get the very small number of very high quality cells that nature would have selected. But we are going to do it with a little device that enables us to find those cells. And in this way, we can ensure that ICSI is not only a very efficient procedure, but also a very safe one.
Now, lastly, the team is also working on a sperm oxidative stress diagnostic tool. Firstly, what is oxidative stress, and why would a tool such as this be important?
Okay, well, oxidative stress is a process which is responsible for a great deal of pathology. We know it's very much responsible for infertility, but it's also responsible for neurodegenerative conditions, cardiovascular conditions, kidney conditions, diabetes. Under oxidative stress is a major pathological mechanism. And what is it? Well, it's quite difficult to understand, but it's basically when proteins and fats in particular, and DNA indeed, in the cells become attacked by oxygen. So if I was to use an everyday example of oxidative stress, I would say, imagine you take a lump of butter and you just leave it out in the air. Over time, it will go rancid. Over time, it will combine with the air, and I guess it's the biological equivalent of rust. It's just combining with oxygen and going off in that sense. Oxidative stress, very important pathological mechanism, underpins a large number of diseases, including infertility. The trouble is we don't know how to measure it. There's a lot of debate and lots of laboratory tests for measuring oxidative stress. Our mission is a bit like our Samson diagnostic, is to develop a very simple one-pot reaction, single reaction that will enable us to capture whether a given system, whether it's a cardiovascular system or a reproductive system, is under this kind of stress. And the reason why you want to do that is that if you can find a patient whose reproduction, for example, is under oxidative stress, then that leads you to a natural course of therapy, which is to use powerful antioxidants. Yeah, the trouble with antioxidants is you don't want to give them to normal people, because if you do, you create another kind of stress called reductive stress. So you don't want that. You only want to give antioxidants to the right people, the people who are suffering from oxidative stress. So this again would revolutionize the practice of antioxidant therapy by giving you a very simple mechanism for diagnosing oxidative stress in your patients or in domestic animals indeed.
And in terms of how advanced that technology is, what are the stages of development?
Yeah, so the first stage there is to come up with a biochemical principle that will enable you to capture a lot of different biochemistry all in one single reaction. And that I think we've managed to do. So we know how to do that in a laboratory. The second stage is now to take that biochemical knowledge and translate it into a device, and engineer a device that people will actually want to use and find easy to use, that will give a readout of the level of oxidative stress in a system. And then the third component is to put that into clinical trial. And again, it doesn't matter if it's a clinical diagnostic lab in a hospital or whether it's the breeding sheds in a cattle farm. The same principle applies. We want devices that are simple and easy to use, that will give the user, whether it's a GP or an animal breeder, the information they're looking for in the simplest and quickest possible way.
Now, it would be remiss of me not to ask you about the Felix device, which, as I said, is firmly on the path to commercialisation and in the latter stages of that commercialisation journey. Now, we can't talk to the data and we can't talk to the results received to date, but the KOL program is advanced and you are getting feedback. Are you pleased with what the KOLs are telling you about their analysis of the Felix device as we move into that commercialisation phase?
Yes, I mean, I guess a sign of how well it's going is that the KOLs that we're working with, who are doing this work voluntarily, we're not paying them to do it, want to do more. They want to, I asked one of the centres to analyse 25 samples to give us feedback on how well the system performed. And at the end of 25, they said, can we do another 25? So, you know, the fact that they have got the confidence that they're willing to spend time evaluating this device gives me confidence that they are finding it useful.
Well, on that note, we will finish up. As I said, the Felix device firmly in the path of commercialisation, but a strong portfolio of research and technologies being developed by yourself, the University of Newcastle team and Memphasys. That portfolio of assets are clearly coming behind Felix, but are very well advanced and fit a very clear market need. So, John, thanks for your time.
Thank you, David. Much appreciated.